So good evening again and welcome back to the Expert Talks, a part of the 9th Karnataka Butterfly and Bee Awareness Week 2026, being organized by the Karnataka Forest Department in association with the Bangalore Butterfly Club. We would like to thank the Karnataka Forest Department, especially DCF Shri B.R. Ramesh, RFO Shri Satita, and the staff of Darsanapalia for arranging for the activities of this awareness week, including these online expert talks. We had a talk earlier in the week on skippers by Mr. Ashok Sengupta. Today's talk is on microbes and butterflies by Dr. Nitin Ravikanthachari. Dr. Nitin is a broadly trained naturalist interested in evolution, ecology, and genomics. His research aims to understand the genetic mechanisms underlying species interactions in response to changing environment. Dr. Nitin completed his PhD under Professor Carol Boggs at the University of South Carolina, where he studied the genetics of butterfly host plant interactions and the genetic mechanisms underlying maladaptive oviposition behavior in Pieris butterflies. He is currently a postdoctoral research associate with Professor Brandon S. Cooper at the University of Montana, studying the genetic basis underlying frequency, transmission, and acquisition of Wolbachia insects, especially Drosophila and Lepidoptera. Dr. Nitin's work on Indian butterflies has taken him to almost all parts of India, including places like the Western Ghats, Kerala, the northeastern states, Western Himalayas, and even the Andaman and Nicobar Islands. His journey has been marked with several achievements. He has authored a much acclaimed research paper titled The Larval Host Plants of Butterflies of the Western Ghats. He is one of the founder members of the Bangalore Butterfly Club, a citizen science initiative to promote the awareness about butterflies, as well as the study and scientific documentation of butterflies in Karnataka. He, along with Dr. Krishna Mekunte, has co-authored the book Butterflies of Bengaluru, as well as a brochure on the butterflies of Bengaluru. Dr. Nitin is joining us today from the US to give us today's expert talk on microbes and butterflies. And viewers, before we start, just a reminder to keep yourself muted and keep your videos off during the session. And if you have any questions, you can drop them in the chat box and they will be answered at the end of the session by the speaker. Thank you. Over to you, Nitin.
Yeah, thank you so much, Priya. That was a very lovely introduction. And hello, all. I'm Nitin, and I'm going to share my screen right now. Let's see if just a second. Is the right screen shown up?
I'm not able to see the screen.
Yeah, now?
Yeah, we can see it.
Yeah. All right, perfect. All right, so welcome everyone, and thank you for being here. I'm Nitin, and I'm currently a postdoctoral research associate in Montana, and I study butterflies, microbes, and So I study butterflies, microbes, and the plants that the butterflies feed on. And today I'll be talking about some of the things that most people ignore when they think about butterflies, and that's the microbes that live inside these beautiful creatures. So for today's talk, I'm going to briefly introduce what microbes are and what are the kind of microbes that live on butterflies and what are the microbes that live inside butterflies and why should we care about studying microbes that are present in butterflies and why microbes matter for butterflies in terms of how they survive and how they interact with nature. So to begin with, what are microbes? If you're not familiar with it, it's something that's microbes are the most common organisms on Earth, more than any other animal or plant that you can think of. And microbes are tiny living organisms, and they're not visible to our eye. You need to see a microbe, you need to use a microscope. And microbe is a catch-all term for a lot of organisms that includes bacteria, fungi, viruses, and other single-celled organisms. And microbes are omnipresent. They're present everywhere in soil, water, on plants, inside plants, animals, and even inside rocks. And they're also present on our skin, inside our body, and everywhere else. And a lot of the diseases that we do get can be caused by microbes. And here are a couple of microbes that occur in butterflies. You don't need to remember the names, but I just want to show you that there's a huge variety of microbes, and they're classified based on the shape that they, the shape and where they occur and what kind of effects they have on the butterflies. So let's go into understanding where microbes live and what they do when it comes to butterflies. So microbes can live on the external surface of the butterflies. It can be either on their body or on their wings. So here is a picture of a nanoscale structure of a common mormon butterfly that you see, which is one of the most common butterflies in India. And this is a picture taken from a butterfly that just eclosed out of a pupa. So it has never experienced the external environment or anything else. But even when they're born, they have microbes on their wings. And this tiny circles here are bacillus bacteria. And these are bacteria that do not do anything on the butterflies, but they're just there as commensal or neutral partners uh, in butterfly bacteria interactions. And this is one of the most common bacteria that you see across all butterflies. And consequently, microbes can also live inside butterflies. So when we think about bacteria living inside butterflies, the first thing anyone thinks is their internal microbiome. So here is a picture of a moth, not a butterfly, but most butterflies and moths do have microbes, and in their gut. So this is where you have where everything that you eat gets processed, and the pink stuff here is the bacteria, and they can contain billions and trillions of bacteria inside their gut, and they do a lot of really important things that I'm going to talk about in a minute. And while we have the microbiome, which is bacteria that live inside your gut but outside your cells, there are a lot of microbes that live inside your cells, inside the cells of a lot of butterflies. And these are known as intracellular bacteria. And this is what I study mostly. So here is a butterfly called the Zerine fritillary. And I photographed this butterfly and then I caught it and then I imaged it under the microscope. And what you see here is the eggs of the butterfly. So the small ones are the developing eggs. That means they're not ready to be laid on the plant yet. And the mature ones are the ones that are going to be laid in the next 1 or 2 days. And the blue color is the DNA of the butterfly, and the orange color is the DNA of the bacteria that is living inside the butterfly's egg. And this butterfly is known as this bacteria is known as Wolbachia, and I'm going to talk a lot about Wolbachia in the future slides. So why should we care about microbes, and why should butterflies care about microbes? Like I said, microbes are everywhere, and they are one of the most common organisms on Earth, and they're present inside butterflies, outside butterflies, and they do a lot of things. And here is a pictorial representation of the kind of things that microbes do and help or either harm butterflies. So one of the major things that microbes do is help butterflies to digest food, or help in producing vitamins that the butterflies themselves cannot produce. So butterflies feed on as caterpillars feed on plants, and while they're feeding on plants, there's a lot of vitamins or minerals and nutrients that are not available to the butterflies, because plant tissues are not nutrient dense, like if you were eating meat or if you're eating another resource. So therefore, a lot of bacteria take up this role where they help the butterflies in digesting food, and a lot of bacteria also produce vitamin B12 and vitamin B2 and other vitamins that are very critical for the development of these caterpillars to become butterflies. So if you were to remove these bacteria from a caterpillar, then they will not survive and they will either not pupate or die very quickly. And the second important thing that these bacteria that live inside butterflies do is protect them against a lot of other kind of disease-causing bacteria or parasites. And the way they do that is, like there is limited real estate or limited space inside inside a butterfly's gut. So when you have really good bacteria just colonizing the entire gut, then you have very little space for the bad bacteria to grow in. And that is how these good bacteria protect bad bacteria from coming on into the gut of the butterflies and protect them from getting sick. And as you know, a lot of butterflies do feed on very toxic plant compounds. Example, just a second.
I request the participants to please keep yourself muted. Yes, Nitin.
Yeah, sorry. So yeah, a lot of butterflies feed on very toxic plants. For example, it could be a crimson rose which feeds on Aristolochia, or your plain tiger or striped tiger which feeds on milkweeds, or your cabbage white butterflies which feeds on cabbage plants. And these are all incredibly toxic plant compounds. And a lot of bacteria that is present inside the gut of the butterfly help in breaking down these plant toxins, making them less toxic so that the butterflies can actually feed on it. So there has been a lot of studies where if you take the bacteria from one butterfly, like for example, if you take the bacteria that is present in the plain tiger butterfly and put it into another butterfly that does not feed on the milkweed plant, the new caterpillar can actually feed on it. So a lot of bacteria do help in breaking down plant toxins. And the last section of microbes are the microbes that influence the reproduction of butterflies. And this is the kind of microbe that I study, and I'll talk to you about this in a lot more detail in a few slides. So these microbes are known as endosymbionts, the purple color here, and these microbes influence what kind of sex is born from in the next generation. So I'm going to talk about it in a few slides. So microbes play a huge role in making sure that the butterflies are healthy, survive, and thrive in the environment. But they can also cause a lot of diseases as well. So in the future slides, I'm going to talk about how the interactions between butterflies and microbes take place in nature. What are some of the microbes that can cause diseases, and what are some of the microbes that can change who gets born and who gets killed when the egg is developing. So the first thing that I want to talk about is microbes that are present on butterflies. And this is a study that was done by an undergrad in the lab. So she was a final year bachelor student, and she did this study, which was published very recently, 2 years ago. And she looked at what are the microbes that are present on the body of the butterflies. And she specifically looked at two different butterflies. One is the silvery blue, the blue butterfly here, and the second one is the Morvon fritillary. So these were the two butterflies that she looked at. And what she did was she went out catching butterflies. And if you remember, during the peak of COVID, where they were swabbing your nostrils or your cheeks, that is exactly what she did. She swabbed the legs of the butterflies, the thorax of the butterflies, and other body parts of the butterflies to see what kind of bacteria was present. And once she finished swabbing, she used to come back and plate it in a nutrient-rich medium, and she would wait for the bacteria to grow. And then she would count how many bacteria were present. So here is, the graph here, you don't need to worry about the details of it, but just, I'm going to walk you through what some of these, red bars mean. So one of the things we scientists do in the field is measure how old a butterfly is when we catch. So we measure that with something called a wing wear. That is, how badly is the wing tone compared to, a fresh butterfly. So a fresh butterfly that is just eclosed gets a score of 1, and the older the butterfly, the more the score is. So wing wear 4 is something that's very old. So what she found was really interesting. So she found that there was more bacteria on butterflies that were intermediate and old. So that means somewhere in the young stages of their life and that were very old. So butterflies that had just eclosed had less number of bacteria. Butterflies that were, in their middle ages had less number of bacteria. But somewhere in their youth and their old age, they had the most amount of bacteria. And not surprisingly, what she also found was that bigger butterflies have more bacteria than smaller butterflies, which would make sense. And she also found that the feet and the proboscis, that, the straw thingy that butterflies use to get nectar, they had more bacteria than other parts of the body, that is the wing or the thorax. And finally, this was surprising, where she found that female butterflies had way more bacteria compared to male butterflies. And this is something that we don't know why that occurs. And this is, something that future studies people can look at in the future studies as to why males carry less number of bacteria compared to the females. So that was the first question that she was interested in. And the second question that she was interested in addressing was to understand, do butterflies actually transfer microbes from when they're feeding from one flower to another flower? So what she did was she designed a really cool experiment where she took a flower, sterilized it, and then got a butterfly to feed on that flower, which is known as a training inflorescence. And then she dissected that flower and again, plated all of the different parts of the flower on nutrient-rich medium, and then looked at what bacteria grew. And then what she did was she took a specific bacteria that is known to glow red, and then she mixed it in salt solution and then sprayed it on the leaf on the flower on the same flower that was used for training. And then what she did was she then put the butterfly on it to count how much bacteria is the butterfly picking up when we know that we have sprayed a specific amount of bacteria on it. And then she did the same thing where she, took the little part of the florets that the butterfly had walked on, plated it, looked at what bacteria grew on it. And then she took another sterile flower and then put the bacteria that had experienced this new red glowing bacteria, and then looked at how much of the bacteria was transferred from flower number 2 to flower number 3. And what she found was that there were differences in what parts of the flower got more bacteria. So donor floret is the flower that the butterfly was first, experiencing, and recipient is where, a recipient floret is where the butterfly was then, put on to see how much of bacteria was transferred from one flower to another. So what she found was that, so before we go into that, I'll explain some of the important structures of the flower. So if you know a flower, it has petals, it has the male part, the female part, and the nectary part. So the nectary part is the one where the butterfly actually puts its proboscis in to sip the nectar. And what she found was that the butterflies actually transferred a lot more, or took a lot more bacteria and transferred a lot more bacteria from these nectaries, and they transferred the least amount of bacteria on the female part. So most on the part that the nectar on, which is not surprising. But when it came to differences between the male and female part of the flower, the butterflies actually transferred a lot less on the female part. So why should we care about what these butterflies are doing in terms of what bacteria are present on the surface of butterflies? Are they transferring any butterflies, and what is the consequences of it? So one of the things that is really important to know is that the kind of nectar that is present in a plant can change based on the bacteria that is present in the plant, so present in the flower. So some flowers have very acidic nectar, some flowers have very alkaline, that is, very like your baking soda kind of a nectar, and some can have lemon juice kind of a nectar. And this matters a lot because if insect likes alkaline or baking soda kind of a taste, then they won't go into flowers that are acidic in taste. So depending upon what kind of bacteria is present in the flower, it can change the entire pH of the flower itself. And when we know that these butterflies can, take a bacteria from some place and then put it into another flower, we know that it can change nectar chemistry and then decide what kind of a pollinator is coming in and actually pollinating that flower. And additionally, they can also spread plant pathogens. So a lot of plant diseases can be spread through just nectaring by butterflies and other pollinators. And it's really crucial to know and keep monitoring what kind of microbes are being spread by different pollinators in your surroundings. And butterflies, when they do these microbial transfer from one flower to another flower, they can, influence, like I said, what kind of microbes are present, and that can influence what kind of pollinators comes. So for example, if there is a bacteria that can kill bees, and these butterflies are moving from one plant to another plant, just, spreading that bacteria, then none of the bees are going to come in, and it's just going to be butterflies that are pollinating it and just cause a whole imbalance or a change in the ecosystem. So this is why I think people should care about what kind of microbes are there on butterfly body parts and what they're doing with these microbes. So moving on, we will talk about another kind of a microbe that is present on the wings of the butterflies, and that can actually cause a lot of uh, issues and can kill butterflies. So you don't have to pronounce the name of the microbe here. It's called Ophiocystis, and in short, we just call it OE. And this is a microbe, which is a single cell. It's not a bacteria, it's a protozoan, and something similar to your malaria. So it's a protozoan. And this is present in a lot of milkweed butterflies. So your plain tiger, your striped tiger, and white tiger back in Northeast, or your classic monarch that is known all over the world. And this is a parasite that can infect butterflies, and it is exclusively found so far only in these milkweed butterflies. And what happens is that, these can form spores that can survive a very long time. And the way this thing spreads between butterflies is that, let's say a female butterfly has this microbe on her body, and when she lays the egg, she also accidentally sprays all the microbes on the eggs. And when the caterpillar hatches and eats the eggshell, which is usually its first meal, the spores break out and they just generate all of these living microbes. And they just keep multiplying and multiplying. And when they finish pupating, the spores can, if spores can then, these bacteria, so not bacteria, these microbes can then form spores again, and then the cycle can repeat and repeat. And what is the consequence of having this microbe on the butterfly's body? So one of the things it does is it reduces the size of the butterfly. So, if you compare a butterfly that is not infected with a butterfly that is infected, infected butterflies are almost always tinier. And that can influence how long you live and how fast you can fly or how long you can fly. And usually females do not, or, not just females, males or female butterflies do not like mating with someone who has smaller body size, who cannot, display their mating behavior with proper flight, and so on and so forth. So having this bacteria is pretty bad. And the way this bacteria spreads is, is when people rear a lot of butterflies in their houses, you crowd them. So if one butterfly has it, if one egg has it, and then, you mix from multiple broods, then you just spread it across broods. And then when you release it out in the wild, then you just keep, spreading more and more of these microbes in the wild, when, that can cause havoc in the entire ecosystem. So here I'm showing you a study that was done very recently based on all the museum samples. So these guys looked at the museum samples collected across the world from 1920 till 2022, and they looked at 5 different species of tiger butterfly. So tiger butterfly from Australia, the plain tiger from all across Africa, Asia, including India and Southeast Asia, and your queen butterfly, and your monarch butterfly, and another butterfly which is very endemic to the islands of the Americas. And here, what they did was they did not go out in the wild and collect butterflies. They looked at the museum specimens, and they specifically looked at how many of these butterflies actually have this microbe. And if let's go into the butterfly that we Indians are very familiar with, which is the plain tiger. So this plain tiger butterfly has this microbe all across the world, all across its habitat range. Fortunately, it was not found in the Indian samples, but this again was an Indian sample that was collected back before the Britishers left the country. So we don't know if the current populations of these butterflies carry this microbe or not. So what they looked at was, trying to understand what is the percentage of butterflies that actually carry this. So the only one closer to us that do carry it is in Sri Lanka, which is about 25% of all the butterflies, all the plain tiger butterflies in Sri Lanka carry these microbe. But the most prevalent butterfly, the butterfly with the most prevalence is your monarch butterfly, because it has been studied for the last 100 years. They have been looking at it every year. They look at how many spores are there in each butterfly and how this affects uh, the monarch butterfly. And here is where I think India is lacking, where we really do not know uh, if the microbe is present in contemporary populations, and how many individuals are actually infected, and what is the consequence of having this microbe. So the two examples that I've given you so far are those that are microbes of microbes that are present on the surface of the butterflies. Next, we'll move on to the microbes that are present inside butterflies, and specifically inside the cells of butterflies. So here are 3 butterflies that I caught, first photographed, caught them, and then imaged them under the microscope to look at this one specific bacteria known as Wolbachia. So the images on the left and right are of a female, and we looked at the bacteria inside the eggs of the female. So again, the yellow, orange, gold is the uh, bacteria, and the blue is the butterfly DNA. So what we do see here is that this butterfly is present in the eggs, the testes, and other structures in the butterfly. So what is Wolbachia? So Wolbachia is a bacteria that live inside insect cells, and it's extremely common. It's known to be present in about 40 to 60% of all insect species. So, by that logic, we can also estimate that about 40 to 60% of all butterfly species have this bacteria. And it's a very special type of bacteria in the sense that it is inherited through the mother. So when just like how we get our mitochondria from our mom, the same way this bacteria is inherited through the mother. The males cannot spread this butterfly; it's only the moms. So because of this bacteria tries to get into the female butterflies as much as possible, and then Wolbachia benefits when it is, when more females are present in the population. So this is a much more, detailed picture of it. So the blue here is the DNA of the female butterfly, and the orange here is the DNA of the microbe. So each single blob of blue is a one single DNA, one single blob of DNA, and you can see for every single cell there is like hundreds and probably thousands of bacterial cells inside the egg. So where does this microbe live? So it mainly lives in the reproductive tissues. So that's in the ovaries and the testes. And sometimes it's also present in other tissues as the gut, in your gut or in the nervous system. And when they are present in the nervous system, they can actually influence a lot of the behavior of the butterfly. So they can influence what kind of temperature the butterfly likes. They can make butterflies do weird things, which is, go and seek out hotter environments than what is, ideal for them, because they survive better in hot climates compared to cold climates. And again, Wolbachia is very difficult to see unless you're using microscopy and DNA sequencing. And even in a regular microscope, you cannot see Wolbachia. And this is a special type of microscopy where we kind of uh, do a lot of chemical treatments to highlight butterfly DNA differently in blue, and then the Wolbachia DNA in yellow. So why is Wolbachia so special? And Wolbachia is one of the few bacteria that can influence reproduction. So what Wolbachia can do is, first, it can kill males. So here in this middle figure here is a phenomenon of male killing, where when a female and a male mate, and if it has Wolbachia and that particular strain causes uh, male killing, only the females are born in the next generation, and all the males are killed. And then it can also lead to feminization. And this is also very common in butterflies, where if a male and a female mate, the females are all born properly, whereas the males, they are feminized. That is, even though they are genetically male, they have all the organs or the reproductive organs of a female, and they act like females, and they can actually lay eggs, and then uh, lay eggs and carry on the next generation. And in some insects, which is, not, which they do not do in butterflies, is parthenogenesis, that is the virgin birth. Whereas, the females, without mating with another male or something, they can just produce females, and they just keep producing as many females as possible. And the most common way in which Wolbachia influences reproduction is through selective killing, which is known as cytoplasmic incompatibility. You don't need to remember the term, but it's something known as selective killing. So it's when, if you, it's a phenomenon where when a male has Wolbachia, denoted in red here, mates with the female without Wolbachia, none of the progeny hatch. So they're all killed. Whereas if a female with Wolbachia mates with either a male with Wolbachia or without Wolbachia, all of her progeny is fine. So what it means is that it's beneficial for a female to have Wolbachia because she can mate with anyone in the population. And it's very harmful for a female to not have Wolbachia because she can only mate with 25 to 50% of the males in the population. And this is how Wolbachia basically makes sure that it increases its abundance and its spread in the population. So one of the most classic examples of Wolbachia in butterflies come from the great egg fly butterfly. So I know the great egg fly here looks a little different than what we find in India, and that is because there's a different subspecies of great egg fly. In India, we have two different subspecies, and as you go south and southeast and far east into Oceania, you have different subspecies of these butterflies. So here, people have been studying this butterfly and the Wolbachia for a very, very long time. So the equator is shown here. So whatever is shown in black is the male killing prevalence. That is how many, if you catch if you catch a butterfly, does it have male killing phenotype or not? And how many of them are actually female? So for example, here in the island of Samoa, you only find females and very few males. And in some of these populations, the Wolbachia is not yet invaded yet. So it can vary very widely between different islands. And on the above the equator here in Hong Kong and Vietnam, you have a different strain of Wolbachia that does not kill the males, but instead causes uh, conditional, killing or selective killing. So within just a single butterfly, you have two different strains which do two different things. And Interestingly, what has happened here is that it has changed how the butterflies behave in the field. So if you were, watching butterflies, wherever, either in JPRF forest or any other place in India, you would see that it's the male that goes and courts the females, right? So the females are basically hanging out at the host plant, and the males come in and they try to impress the females. And then, if the female likes, then she mates with the male. Whereas here, because the males are so rare, you see that a single male is being courted by 10 to 15 females at a time. So it's a complete reversal of butterfly behavior itself. And one of the things that also is very prevalent here is that even though so many females are courting the males seem very uninterested in mating. So populations here in these islands are a lot less, not because of any other factor, because of this particular male killing Wolbachia. And very interestingly, what has happened in the last 100 years, and this is why I always like, encouraging museum collections because of the kind of things you can learn from it. So this butterfly, uh, the great egg fly had this particular male killing Wolbachia. So when you have a very bad thing happening in the population, evolution usually overcomes it because of mutation or some other things. And what happened here is that from 1920 to 2000, the male killing phenotype was there, and only females were born most of the time. But suddenly there was a mutation in the butterfly, and that made the Wolbachia inactive. So basically, it suppressed all of the Wolbachia effects. So you had 50-50, 1:1 ratio of butterflies, of males and females that were being born after 2000. And between 2005 to 2020, what happened was that a phage is nothing but a virus that infects bacteria. So somehow this virus got into the butterfly and then got into the Wolbachia, and then it transferred its DNA into the Wolbachia. And then what happened was that when this uh, suppressor was there, which restored 1:1 of male and female ratio, that was lost again. Because then the Wolbachia evolved a new thing, and now you have male killing phenotype again. So within a period of 150 years or 100 years, between 1920 to 2020, you had a male killing phenotype where, butterflies were only females that were born. And then that got reversed because the butterflies evolved to suppress that male killing. And because of a third partner, which is a virus that infects bacteria, these bacteria were able to re-evolve this particular male killing phenotype. And now they're again back to being female based. So and the really unfortunate thing is we don't know what is happening with Indian butterflies. We do see, males and females pretty commonly in some of the urban areas where we look at butterflies, but we do not know how it is through the entire country. And I'm pretty sure that a good amount of these populations might have either the selective killing phenotype of Wolbachia or the male killing phenotype. We just don't know. And there are other examples of really cool male killing bacteria in butterflies, that is Wolbachia in butterflies. One is the Acrea ensedon, which is a relative of tonic coaster that is found in Australia. Here again, it's like some populations have only 100% females, and then some populations have like over like 80 to 85% of females and very few males. And another butterfly that is incredibly common is our grass yellows. So both, like this, the first study was conducted in the Japanese grass yellow Urema mandarina, and they found that there is this male killing. And this butterfly also has three other different strains which do three different things. And in our own common grass yellow, this is incredibly prevalent as well. So if there's anyone who's interested in looking at Wolbachia in butterflies, I don't think there's a better butterfly than the common grass yellow in India to look at, because they're so common and they're very prevalent, and they're incredibly easy to, look at how many males are born from a single egg batch versus how many females are born, and is there a male killing phenotype or is there some other phenotype? So what is the use of studying these sex ratio distortion? And we know that population growth depends on female numbers, because you have more females, more eggs are laid, and then you have higher number of offspring. But if you have very biased female sex ratio, that means you really do not have enough males to mate with the females. And if you have one male mating with 99 females, that means the genetic diversity can drastically decrease. And when you have decreased genetic diversity, it can then lead to, being susceptible to new diseases and not being able to adapt to new challenges. And this is the way where a single tiny microbe, which is not even visible to our naked eye, can drive rapid change in evolution of butterflies and cause so many different behavioral changes. So now, a lot of people can question what is the point of us studying these things in butterflies if we really cannot apply this to our own human life and our own human, health and other aspects. And there's a really good reason as to why we should study organisms like Wolbachia. Because Wolbachia is a bacteria that can proliferate very, densely inside a lot of their, host tissues, like, either butterfly or other organisms. What it also is known to do is that when a butterfly or another insect has Wolbachia, it is resistant to a lot of pathogens. So this is what people, when Wolbachia was discovered in 1920s, people thought, oh, this can be used for some other application that we humans can benefit from. And we know that mosquitoes are one of the worst organisms on Earth for humans because they cause more disease than any other insect. And these carry chikungunya, Zika, dengue, and all of these other, and malaria. So what people did was that they took this Wolbachia that was present in the fruit fly, in your vinegar fly that's all around your, fruits and everything. And they took that Wolbachia and they put it inside this mosquito where Wolbachia really does not occur. And what they found was that when the mosquitoes had Wolbachia, it really did not affect their population, but it absolutely killed all of the viruses and other disease-causing pathogens in these mosquitoes. And this can be a very important tool to fight mosquito-borne diseases, especially in a country like India, where we have more than 500,000 to like a lakh cases every year of dengue or chikungunya or malaria. So a timeline of how this was done. So Wolbachia was first identified in 1924, and then based on a lot of laboratory experiment, scientists said that we can use this as a way to control mosquitoes and mosquito populations, or make sure that mosquitoes don't spread diseases. And what we do now is, collect mosquito eggs and then under the microscope inject Wolbachia into these eggs. And then when we release these mosquitoes in the wild, they do not have these other pathogens such as dengue or chikungunya. And the more they mate with the wild mosquitoes, the population of dengue and every other pathogen keeps decreasing. So in Australia, they tried this out, and within 3 years, there was a 98% reduction in Zika virus that was reported in human beings. So it's a very, very important tool. It is way better than a lot of the tools that we currently use, which is, spraying incredibly bad chemicals like DDT or, what other airborne chemicals that we spray in our cities. So to end this talk, I hope I've convinced you that microbes are incredibly important for butterflies because microbes interact with every stage of the butterfly's life, and they influence not just butterflies, but also all of the other pollinators around us. And microbes influence the health of a butterflies. And finally, microbes can influence long-term evolution by manipulating sex ratios. So to end my talk, butterflies are not just individuals, they are ecosystems because they contain their own microbiome outside of their body, inside of their body, inside their cells. And these microbes can be helpful to them, or they can cause harm, or they can basically be puppet masters where they control what a butterfly does. And to understand butterfly behavior, to understand what butterflies do and everything else, we need to understand this hidden partner to really get a sense of uh, the what the role butterflies plays in the environment, and it's really important that people start paying attention. and I'll be happy to take any questions you have right now.
Yes, Nitin. So we do have a few questions. Uh, a question from question from Indumati. Does Wolbachia live on butterflies alone or other insects as well? You just
Yeah, they live in, I mean, if you,
do
I would say about 40 to 60% of all insects carry Wolbachia, and we really do not know the true prevalence. And one of the things that I'm doing is looking at Wolbachia in butterflies, but I'm also collecting other insects such as grasshoppers and flies and everything else that I can get my hands on to see what is the prevalence of Wolbachia. And what we do know, at least from studies done in 2000 and 2000, between 2000 to 2010, is that a huge amount of insects have Wolbachia, and they're much more prevalent in tropical regions compared to temperate regions. Temperate in the sense, in the US or in Europe, in temperate Europe. They're much more prevalent in tropical and subtropical countries like India, Australia, Africa, and other regions.
Okay, the next question Vishal has asked, does it mean if Wolbachia is prevalent in most butterflies in the local area, the local population could end up without males after a few generations?
Yes, so here is the thing. So one of the things about butterflies is that they can travel really long distances. So let's assume that you have a small population where this male killing Wolbachia is there, and the next generation, all you get is females. But the females will try to find males, or the males can travel long distances from a different population, come and mate with the females, and just keep these male killing phenotype going on and on. And over a period of time, like what we saw with the great eggfly butterfly, is that these butterflies themselves can evolve a way to suppress the effects of this male killing. So it can be both. So, it can be a combination of getting new males every generation versus you evolving a way to suppress this bacteria over hundreds of generations.
A question from Navami, do bees have Wolbachia?
Yes, bees have a lot of, like a lot of bees have Wolbachia, and that is something that people have actually looked at. I mean, that was one of the most studied organisms. And in bees, Wolbachia don't just, they don't actually, influence sex ratio, but one of the things that they actually do is provide them with vitamin B12. So if you clear bees of Wolbachia, they actually do not do really well. So in bees, Wolbachia is really important.
Indu has asked if there's any relationship between Wolbachia presence and gynandromorphism in butterflies.
Yes, that's actually a great question. And a lot of the gynandromorphs, so gynandromorphs in butterflies can come about in two ways. So gynandromorph is where half of the butterfly is female, half of the butterfly is male, and it can come about by two mechanisms. One is, during sex determination, which is when the egg is developing, you can have some, issues with sex determination where there is not, where all the cells do not get the same information, and then you have half female, half male. And the second thing that can happen is that when cell division is occurring during the egg development, if Wolbachia just goes into one set of cells and not another set of cells, you can get gynandromorph. And this has actually been shown in the butterfly known as bicyclers in Africa. And the closest relative to bicyclers is your bush browns. So it has actually been shown in some of these butterflies that having Wolbachia can increase the incidence of gynandromorphism.
Vishal has asked if the microbes change for a caterpillar versus the butterfly.
So yes, so there has been a couple of studies which have looked at, what is the microbes that are present in caterpillars, what is the microbes that are present in pupae, and what is the microbes that are present in adults. So let me see if I can share my screen again, because I think that will be. So here I made here is a thing where I talk, where they show transient gut microbes. So this is, microbes that are only present in a very specific part of the life of the butterfly. So yellow here is the resident microbiomes, which basically stay throughout their entire life. So there are some amount of bacteria that are present in across caterpillars, pupae, and butterflies. But there are some bacteria that are only present when the butterfly is a caterpillar because it helps them feed on the uh, plants. And then when they become pupae, those are reorganized, and there's nothing. Those bacteria just die or they don't make it. And then when they come out as butterflies, there's a completely different set of transient gut microbes.
Kiran, okay. Yes, Kiran wants to unmute and ask a question. You can go ahead, Kiran.
Hi, thank you, madam. Thanks, Nitin. This is something very unique. I have attended many webinars on butterfly, but the first time a webinar about the microbes. I never thought of microbes and that connection between butterflies, especially in the gut bacteria. My question is specifically on the gut bacteria, the stomach-friendly bacteria, which is helping them to digest the food and all. So, in higher organisms like humans, and I heard that this is this the first colony of the bacteria getting into the stomach just soon after the birth, when the embryo at the time of birth and soon after the birth. But butterflies being egg-laying organisms, and when the gut bacteria is entering into the first colony, entering into the gut, and the second thing is during the metamorphosis, what is happening? Like the larval gut and the butterfly gut, it is totally a kind of mash up, right, in the at the time of inside the pupa. So what is happening to these bacteria? It is totally different species of bacteria after they evolved as butterflies, or they are same at the time of larval stage and the adult.
So yeah, to
Thank
answer
you.
your first question, I mean, since a lot of these butterflies do get the way they reproduce is through laying eggs. And what is the first thing that the caterpillar feeds on? It's the egg case. So the egg case is where they get most of their bacteria from. So one of the things that happens is that when the females lay eggs, they a lot of the bacteria when they enter or when they are coated on the surface of the eggs, they get into spores, and spores are very durable. They just don't get destroyed, and when they get ingested by the butterfly, that's when the cell wall breaks, and then you can then those proliferate inside the gut of these caterpillars. So yeah, that is how a lot of butterflies or a lot of egg laying insects get their first set of gut microbes. And yes, so like I said, there is a particular set of microbes that are only present in the caterpillar stage, and when you become a pupa, then they can get reorganized, and the way they get reorganized is very can be simple or complicated, but the most common way in which they get reorganized is basically in that particular environment in a pupa where the pH of the butterfly changes, the environment changes, what bacteria can actually grow in that environment. If a particular bacteria can grow really well in that environment, those make it to the adult stage. If a bacteria cannot grow in that particular environment, they just get lost between caterpillars and adults.
Thank you. This is totally eye-opening. Thank you.
I'm glad you enjoyed it.
Yes, of course.
Okay,
Could,
we have.
could Wolbachia create a new species by breeding through different types of butterflies?
Yes. So yeah, so there is a huge avenue of research which talk about speciation through Wolbachia. And I mean, it's a very complicated way to actually say this is a new species versus that's a new species. But what we do know is looking back through evolution, like a lot of these butterflies are at least 10 to 15 million years old in the evolutionary tree. And we can say, okay, fine, if there was a at least maybe not in butterflies, we still don't know, we still don't have that evidence because not a lot of people have looked at Wolbachia and butterflies. But in fruit flies, especially in Africa, people have shown that being associated with a different, when there were ancestors, you had a single type of Wolbachia and then there was some changes that occurred in Wolbachia, and then they got split, and that actually led to speciation and formation of two different species which cannot mate within each other now. So it has been shown that yes, Wolbachia can lead to speciation, but we need to look at more examples with butterflies because Wolbachia and butterflies is still a very emerging field.
We have the next question from Minoti, asking if closely related insects share the same type of microbes.
They actually don't. So surprisingly, they don't. And that is because, so it all depends on what kind of food they're feeding on. So sometimes they can share the same kind of microbes in case of milkweed butterflies because they all feed on milkweeds. But for example, if you look at something like brush-fruited butterflies, the nymphalids, which feed on so many different, even close-related organisms feed on entirely different host plants, they can have completely different microbiomes. So it all depends on more than closely related from an evolutionary perspective, how closely are their food, plant, and their behavior similar.
Can Wolbachia found in humans and other animals?
Not in humans. So it's exclusively present only in insects and some nematode worms. So nematode worms in the sense roundworm and your other worms that are like for example, if you have a pet, your pet gets worms, right? So cats or dogs, they are present in those. And one of the easiest ways to cure them, if a anti worm thing is not working, the next thing to do is to just use antibiotics specifically targeted at Wolbachia on these parasites. So it's only present in two different types of organisms, the parasitic worms and insects.
What about the endangered species? They're like, they would get like, they would die, like they would end, their species would end fast if Wolbachia continues. Like they won't have anybody to mate with.
So yeah, that is a thing that people have started to look at is to see, what proportion of endangered species do have Wolbachia. And while, like for example, I know at least in the US, there have been reports of some of the endangered species that have Wolbachia and they are closely monitoring their population. And this is where, a lot of our own intervention comes into picture. So, thousands of years ago, even if, I don't think anything was endangered, or even if they were endangered, they had a lot more larger population sizes. So even if there's male killing in one population, another population can keep, getting a male from another population can keep coming in and mating with this male killing population here. Now, because of, whatever changes we have made to destroy environments. So this is where I think we as like the public, the scientists and everyone, we can look at, okay, fine, if this endangered butterfly, if this butterfly is actually endangered, and if it is endangered, does it have the male killing phenotype or does it have the selective killing phenotype? And if it has the male killing, then you keep monitoring it, then you keep getting males from a different population, if it's not connected, and make sure that they survive for the next, foreseeable future.
Manjusha, we have a question. Is there any chance that Wolbachia can become an organelle like mitochondria after many years of evolution?
Yeah, that's actually, we call this the symbiosis continuum. So symbiosis means something that lives inside an organism, something that is interacting with an organism and it is beneficial to both the interacting partners. And what we do know is that Wolbachia, like the way mitochondria became an organelle is because of symbiosis. It came, it got engulfed and then it, gave a bunch of beneficial things to the cell, and the cell in turn gave something beneficial to the mitochondria. So mitochondria is billions of years old, whereas Wolbachia is not a billions of years old. Most insects got their Wolbachia between the last 5,000 to 15,000 or maybe 20,000 years. We are still, kind of understanding when did specific insects actually get their Wolbachia. And what we do know with a lot of symbiosis is that over a period of time, most of these parasitic, relationships evolve into become mutualisms, and that's the only way they get to be there long term. So maybe in a billion years, if Wolbachia is still, with the same insect, then they might become, or, across the tree of life, they can become an organelle. But what we do know about Wolbachia is that it can switch from one different butterfly to a different butterfly. So they don't really stay in a single butterfly for a very long time on an evolutionary scale, maybe 30,000 years, maybe 40,000 years, but after that they switch. So there's not enough time for it to become an organelle.
So Navami is also asking, could Wolbachia create a new species by breeding through different types of butterflies?
I mean, not through different types of butterflies, but when a single butterfly, for example, you have your grass yellow and they have multiple strains of Wolbachia. So, because of some mutation or something else, if one strain of Wolbachia kills the other strain of, Wolbachia, or, when butterflies with two different strains mate, they don't produce any offspring, then that can lead to speciation over a period of time.
She's also asking, can global warming affect Wolbachia?
Yeah, actually, like we know that Wolbachia survive really well in ideal temperatures. At least, with fruit flies, we know that they are best transmitted from mom to offspring between 23 degrees centigrade to 29 degrees centigrade. So below that, they don't do really well. And above that, because of heat stress, they don't do really well. So over a period of, because of global warming, you can see uh, decreases in Wolbachia at very high temperatures, or in areas with very high temperatures. And that's also the reason. So like if you remember, I said that Wolbachia can change the behavior of butterflies. So when a Wolbachia is incredibly packed inside of the brain of a butterfly, they really cannot, they can influence butterfly behavior where they try to start seeking out hotter and hotter places. So that is to make sure that they can get transmitted well. So if they get really hot, then they just die.
So I have a question, Nitin. You had said that they are found more on the smaller butterflies than the bigger. You mean the smaller size means which is kind of usually that happens when they are malnourished, or isn't that what you meant? Of the same
That's with
species. So on
that is
smaller
with
size in
the
the same species.
that is with the milkweed butterflies and the parasite, the OE parasite. So having that parasite makes it very, makes you very tiny. And not, yeah, Wolbachia has nothing to do with size differences yet. I mean, we have not really looked at that in detail.
So is that like in humans, if somebody is malnourished, they pick up more disease,
Yeah,
their
yeah,
immunity is lesser down
yeah,
and they pick up. So is it
yeah,
like that it picks
yeah,
it
it's
up
exactly
because
like
it's
that. Yeah.
smaller because it's malnourished, right?
Yeah, yeah. So yeah, I mean, a lot of things are, similar in insects, plants, and as humans. Is when you are malnourished, you are susceptible to a lot of things. Your immune priming is less. You cannot fight off diseases, and you are susceptible to things that are not that other people who are, healthy can not have any symptoms at all.
Okay, thanks. So Navmi has another question. So there are no butterflies in the colder regions of Russia and Switzerland?
There are, butterflies are all across except in Antarctica. And so some of these butterflies in Russia or Switzerland or even in Montana, where, right now it's minus 13 degrees centigrade, we do have butterflies in the summer. So what happens is that when in all of these colder regions, the butterflies enter a period of hibernation or diapause where they just, stay dormant for all of the winter months. So during that time, the Wolbachia also stays dormant inside the butterfly. And depending upon the temperature during the summer, they just, reproduce, they get onto the next offspring, next generation, and then they just become dormant the previous cycle. So this cycle keeps going on.
So as soon as the conditions become favorable,
Yeah,
they again get
yeah,
activated.
yeah.
Okay, we have a question from Austin. Are Wolbachia subject to things like bacteriophages, and do arthropods have any self-defense mechanisms against these?
So yes, like I said, so one of the example I gave you was of the great eggfly butterfly, which basically ingested a like a population that ingested a bacteriophage that became common, and that went and infected the Wolbachia, and that restored male killing, which had gone from the population. So yeah, a lot of, one of the really interesting thing is that in the genome of Wolbachia, you have two parts. One is the core genome, which is all Wolbachia, and the other one is the mobile element, which is basically whatever Wolbachia has picked up from other organisms like bacteriophages or other bacteria from the insect itself. So this mobile element keeps changing. It's very rapid. It's very, within a few tens of or hundreds of generations, they can change. And all of this is made up of bacteriophage particles. So it's very common.
Devraj has asked a question. If we take two similar species from two different altitudes, does the altitudinal range cause any behavioral change in butterflies? And same time, the same kind of Wolbachia will be present in similar species that live in different altitudinal range.
I mean, so this is a classic thing that a lot of biologists do with common garden experiment, is where you bring, butterflies or plants or whatever from different altitudes, different elevation, different conditions. And then if you grow, if you breed them in the lab, you can see differences that are very specific to, you can dissect what is contributed by the altitudinal adaptation versus what is, not uh, what is contributed by the altitude and what is just plastic in the sense that can change a lot. And what was the second? Yeah, and yeah. So if a Wolbachia is there in both the populations, they can have the same strain of butterflies, or they can have different strains of butterflies. So it all depends. Unless you look into it, you do not know if it's the same strain or not. So one really good example is the fruit fly or the vinegar fly, which is present in all the continents except Antarctica. And then we know that across all of these populations, there's just one strain of Wolbachia, which was which basically spread in the last 4,000 years.
Okay, now he's asking, could I ask you what got you into deeper studies of butterflies?
I just wanted to impress a girl, and that's what got me into butterflies. And yeah.
That's a really simple answer.
Yeah.
Anyway, so yeah, I think most of the questions. Yeah, anybody would like to ask any question, you can unmute and ask. Okay, we have one more question from Manjusha. Are the genes that influence butterfly characters present in plasmid DNA or the nucleoid?
Let me see that question.
The last question in the chat.
Yeah, so Wolbachia do not have a plasmid. It's just, a single circular, simple circular genome with, UTR on one side and the end on the other side. So it's only on the nucleus that's where this, genes are present. And yeah, there's no plasmid in Wolbachia.
Okay. You had said that the mosquitoes, if you have them infected with the Wolbachia, the diseases in them, what is seen as they're much reduced levels.
Yeah.
But do the mosquitoes transfer the Wolbachia to other organisms that they touch?
Not other organisms. So since it's strictly transmitted from moms to offspring, they only keep, transmitting from, generation 1 to generation 2 to generation 3.
So it's only internal and
Yeah.
through the offspring.
Yeah, and the other thing,
like in other,
yeah. So, if it was like the perfect thing, what you would have to do is just, infect the mosquitoes once, and then you can eradicate dengue or Zika virus. But the problem here, at least with the Wolbachia variants that they use, is that they get lost from the population of mosquitoes over a few generations. So you need to keep, injecting and releasing these flies. And that's one of the things that, we are trying to understand, is to find better variants that are stable across a long period of time till we can completely suppress Zika or dengue or malaria. And that's the whole reason, I started my postdoc with studying butterfly Wolbachia, because every Wolbachia that we use in mosquito control or other, insect vector control is from the fruit flies. And these fruit flies, while they are present all across, they are very sensitive to temperature and other things. Whereas we know that some butterflies, for example, if you look at a butterfly in India, something like chocolate pansy or something, they are present literally in the hottest of environments. They do really fine. So if something like that has Wolbachia, then you can transfer that into our own local population once and for all, and probably 10 years you don't have to worry about it. So yeah.
So you're saying that they don't last for long and you have to reinfect them. So is that
Yeah.
because the Wolbachia don't find the mosquito as a suitable environment, or is it because the mosquito's own immunity or ability to evolve
It's a
over this is
combination of both. It's a combination of both.
okay?
So mosquitoes do not. I mean, not all. Some mosquitoes have Wolbachia naturally, but a lot of mosquitoes, especially the disease-causing ones, they do not have Wolbachia naturally. So you need to have you need to infect it with a Wolbachia that is incredibly strong and incredibly adaptive, which can adapt very quickly to the mosquito's internal environment and can also overcome all the defenses that mosquitoes have in terms of saying, oh, we want to clear it as soon as possible.
So both things happen to
Yeah.
reduce them.
Yeah,
Okay.
everything is, complicated when it comes to real world biology.
And Naomi has a fun fact to share. She says, "Did you know Karnataka government textbooks have chapters on butterflies?"
Oh, that's really nice. I'm
Okay.
glad we didn't have it when we were, when I was in school. So
Okay, I think we have done with most of the questions. Does anybody have any other questions? You can unmute and ask. Otherwise, we can wind up. Anybody would like to ask a question? I think something has come. No, just thank you. We'll just wait for a second. I think we're done with the questions, Nitin. So thank you, Nitin, for
no problem.
this
Yeah.
really wonderful talk. And it's like, you when the topic came, microbes and butterflies, I'm sure many people thought, I mean, it's a totally biology kind of subject, but you make it sound very down to the beginner's level to be able to understand. That is the uniqueness of your talks, that you are able to make everybody understand what you're talking about. Thank you so much for this lovely session. And I learned a lot, and I'm sure the viewers will also agree with that same way. And viewers, before we sign off, the recordings of these talks will be available on the Bangalore Butterfly Club channel of YouTube. And even the previous year's recordings of all the talks are also available there. So please do visit the channel and take a look at the different topics on butterflies and other topics which are available from the previous year's butterfly and bee festivals and awareness weeks. Yes, Nitin, thank you again. And thank you to the Karnataka Forest Department too. Thank you all for being here, viewers. And we'll just a minute, Nitin. We have a few, I'll just check a few messages have come in. No, they're all thank you messages for you. No more questions. So thank you. And I think we will sign off now.